So the very fundamental idea that the immune system can kill cancer has been debated for over a century. However, in the last 20 to 30 years, with the discovery of dendritic cells and with the discovery of tumor antigens, the role played by the immune system has been more clearly established. And today, the immune system is actually viewed as a personalized killing machine that can kill cancer really well, remarkably, 99.99% of the time. And cancer is that one cell that has escaped destruction. It is that 0.0001% of the time that the immune system has failed. But going back to the question, can the immune system be deployed to kill cancer? Yes, there are different approaches that can be used. And of all these approaches, cancer vaccination definitely represents one very successful approach. So if you think of vaccines, vaccination has already eradicated 26 infectious diseases thus far. So this success, coupled with our very deep understanding of cancer, of the immune system, actually opens up the possibility that we can use vaccine-based interventions to prevent or control cancer. And our success with vaccination is primary prevention for hepatitis B-associated hepatocellular carcinoma and human papillomavirus associated cervical cancer represents a giant step in the right direction. And echoing this idea, the National Cancer Moonshot will support cutting-edge technologies and opportunities in vaccine development to prevent cancers. So there are two broad kinds of cancer vaccines. There are the preventive vaccines that are applied to prevent cancer, and there are therapeutic vaccines that are applied to treat cancer by strengthening the immune system's defense. Now, going back to preventive vaccines, there are different kinds of preventive vaccines. And if you think about cancer, it evolves over time. And as this occurs, there are pathological changes that become irreversible. So the aim of preventive vaccine is to really stop this progression. And so when you think of prevention, there's primary prevention, which is really applied before the initiating event. For instance, vaccinating children with Gardasil or Cervarix against human papillomavirus infection is an example of primary prevention. Secondary prevention refers to interventions that are applied at an early stage to actually prevent progression to invasive cancer. For example, intervention in DCIS to prevent invasive disease or localized prostate cancer to prevent invasive disease are examples. And effective screening programs are a very important aspect of secondary prevention. Tertiary prevention really refers to strategies to prevent recurrence of disease after curative cancer therapy. So today we have a deep understanding of the tumor microenvironment, and this, coupled with advances in sequencing, will allow systematic approaches for whole genome profiling of even pre-malignant lesions. This will not only lead to early detection, but it can also open up opportunities for early intervention. And newer adjuvants, newer delivery systems, and vectors would actually improve efficacy and safety of vaccines. In addition, the remarkable growth that we are seeing in immunotherapy can actually help move immunoprevention forward. For instance, therapeutic vaccines could be translated into preventive strategies provided there is good safety data and also the targeted antigen is expressed in the early lesion. There is plenty of evidence to support the idea that early intervention can be efficacious because the immune system is still functional, whereas time and again we see that therapeutic vaccines fail because the immune system is dysfunctional.